Yes.
Okay.
I think. So, good afternoon, everyone. Today, I'm honored to introduce Professor Jan Dincer. His research interest is in the development of bioanalytical materials, sensors, or microsystems, and their combination with data science. Today, I'm honored to introduce Professor Jan Dincer. So we have the echo. Okay. Sensors or microsystems and the combination with data science and artificial intelligence for One Health, human and animal health and environment. His focus is on disposable sensors technology for point-of-need and wearable applications. He's an editor of Biosensors and Bioelectronics. He's got many awards. For example, he's one of the top 2% scientists by Stanford Elsevier rank. He's got the Biosensors and Bioelectronics Best Paper Award and in 2021. But of course, he will introduce his career in a briefly way. So please welcome Professor Jan Dincer.
Thank you very much, Adam, for this nice introduction, and thank you very much, everyone, for coming. I'm happy to be here. So today I'm going to speak about our work at TUM, at Technical University of Munich. And so I would start just a brief, so after Adam's, I don't need to go in. So I was born in Turkey, which was asked. I came in 2002 to Germany, then did my PhD and studied there, PhD at the University of Freiburg. Then I was 2 years in visiting researcher at Imperial College London. At the same time, I had my group again in Freiburg at the Center FIT. And since last year, October, I'm a member of a TUM. So that's just a short introduction of mine. Today I'm going to speak about the technologies that we are developing, but I would like to start with a motivation. So why we are interested in such a sensors in next generation diagnostics. And one of the visions that we have is currently we are living in a population-centered care, which means actually if you got a drug subscribed, then you have a prescription which is tested on a small cohort of a patient, telling you that these doses should be right for you. This is in some cases, of course, optimal, but in many cases, it's not actually fitting to your physical conditions. And what we need, as this case also show, that we need a patient-centered care. And similarly, for the sensors, we are developing sensors, and once we have a system, we are looking what type of analytes we can measure with this. However, what we also need to consider is what is the requirements of these patients, and we need to engage with the patients in the very early stage to understand their needs, because acceptance is a very important point. We will talk about today wearables. In this case, we need to convince people about why they should carry the sensor systems that we develop, and this is one thing if we need to involve them in the very early stage. The second point is we are still living in a sick care. The diagnostic starts once either your doctor has a suspicion on a disease or you are really sick, and then we need to monitor your parameters. And however, with the upcoming devices on the wearables, what we see is actually we can monitor a lot more than we can ages ago. And this allows us to realize a proactive healthcare. So you, as a user, as a patient, you can monitor your data, you can adapt your lifestyle, and you have the power on yourself and control it. And so this is a cover from 21 from my group. And for this approach, of course, we need devices. So here on one side, we see a patient lying in a hospital. We are monitoring different parameters. And nowadays, we are working on wearables to realize this monitoring before you are having such a clinical setting, but actually in your real life without disturbing your daily life. So that's kind of the idea behind. And how we would like to approach this, of course, everything starts with materials. In my group, we do not develop new materials, but we functionalize them. So here, an approach actually showing how we use silicium-based gold nanoparticles to create hydrophilic electrodes, in this case, in this cover shows. And the second thing, of course, once we have the materials, the sensor materials, we need to talk about the biomaterials, biorecognition elements. And for this, over the last years, we have a nice approach by having the help of the synthetic biology, like we can use CRISPR effectors as diagnostic tools, or binding proteins from the resistant bacteria to detect actually antibiotics, for example, in this case shows. So we, having the materials, we have the biorecognition elements, we can sense. So the next step is, of course, to understand the data. And here, of course, the artificial intelligence and data-driven methods comes into the play. And here in my group, so we are at the beginning, so we are learning these approaches and we are looking how we can integrate these technologies into the sensors. And of course, we need to close the loop. So the last idea that we had and also talk about before is how we can assure the data safety. So because we are monitoring and getting, let's say, every 20 milliseconds one data. And we need to first understand it and then second secure it. And there, for example, we might use ledger-based technologies like blockchain to close the loop. So, for example, having your own blockchain for your health, where you give access to the doctors, to the insurance if you want. And so if a wearable reports something, the doctor gets noticed, and in case of a drug prescription, he starts a smart contract, orders the drug, the insurance will inform and will pay it by cryptocurrencies. So this is kind of course, here we are not active at all. This is just in the thinking stage, but just to have a roadmap about for the future of our devices that we develop. And of course, all we do is creating biomedical sensors and wearable sensors. So here you can see two different examples which we did in my group over the last years, which I'm going to speak today also about them. First of all, I would like to give a short overview of what we are doing. One area which we are working on is multiplexing. So we are trying to detect multiple analytes or same analyte in different samples simultaneously. Then with the same chip, we have a crazy project where we are trying to use these polymer chips on a living animals to create a lab on a bird approach, which I will speak later on. Before the pandemic, we had the idea to use paper for paper-based exhaled breath analysis for different continuous and non-invasive monitoring. And finally, the latest approach which we had is an OptoAssay, where we use optogenetic switches, which can bind and release by changing only the wavelength to create a microfluidic free and pump free assay, which I will show you in the end of my talk. So apparently it's talking at 1 o'clock in Europe. I'm a little bit slower while speaking, but so starting with the more matured approach. So this is the microfluidic multiplex biosensors. So we can detect simultaneously different analytes or same analyte in different matrices. We can go up to 8 parameters, and our current targets are antibiotics, inflammation markers, nucleic acid, and hormones. So here you can see some of the microfluidic previous versions which we use. And actually, my plan is to go with application to application. And our first application which we are using with these types of sensors is therapeutic drug monitoring of antibiotics. Here, if you need to monitor a drug, the road is you collect the sample, send the sample, and the measurement is done by chromatography or immunoassays. And especially for antibiotics, it's not easy to find laboratories which are specialized for it. And in the case, a day later, for example, when I was in Freiburg, the next laboratory was in Dortmund. So we are talking about, I think, 800 kilometers from each other. So the next day you get the results. The doctor can decide if the doses is correct, can adapt, and apply it a day later. So this is normally it's okay. However, in the life treating cases like sepsis, a day is too long. So you need to have 3 days to adapt and to have this therapy correctly to save your patient's life. And for this, we propose an approach which we call personalized antibiotherapy, where we would like to measure the antibiotics for managing the therapy. Then we are measuring the inflammation markers by measuring to assess the therapy success. And finally, we would like to also prognose organ functionality, like kidney monitoring. In the first work which we did in this project, we used different pig samples where we underdosed, normalized, and overdosed. And we collect different samples, except bread condensate, plasma, whole blood, urine, and saliva. And just to explain you easily how our sensor is built, so this is, I think, the simple version is easy. So I'm coming from electrochemical background, and we do not want to steal or put anything on our working electrode and actually has a problem with this electrocatalytic activity. So the idea is we define in a microfluidic channel an area which we call the immobilization area and put all of the assay parts. So, and once you put the drop, it runs here until the stopping barrier, which is a hydrophobic barrier. And once the immobilization is done, you do suck it from the same way so that your electrodes does not have any control and contact with it. So, and final measurement done, of course, through the barrier by closing everything. So with this manner, you can work all type of different samples and you do not harm your working electrode at all. The assay that we use is penicillin binding protein, which just recognizes the beta-lactam ring of a beta-lactam, which is actually a little bit problematic because biorecognition elements needs to be very specific. However, beta-lactam ring is defined for all beta-lactams, which means it's unspecific, and you can measure with this assay all different beta-lactams. The good thing is, usually if you apply an antibiotic, you know which one you gave your patient. So it's, in that case, even better because you have one assay, you know the antibiotic that you gave patient, and you can use always the same competitive assay to measure your beta-lactam antibiotics. The highlight of this work was that we could, for the first time, detect also antibiotics in exhaled bread condensate. So, and my PhD student won one of the prestigious awards in Germany, CLE Award in 2022. Let's look at the results. So we had two aims, just to make a whole blood measurement like the glucose measurement that you can do with fingerprint. Can we do the same for antibiotics? Yes, we can. So you can see for all cases, we see nicely after the antibiotics, the signal is going down, and after 4 hours, it comes to the initial state. So we can measure antibiotics, track over 4 hours. And again, that was the first thing. And in the second case, we had worked with EBC. So this is the exhaled bread condensate, which we get it from the animals. And at that time, you could see residues, but for the first time, we could really track EBC, in EBC, the antibiotic concentration, which opened up, of course, that you can measure antibiotics from exhaled bread. So this is, of course, with the pandemics and also with the masks idea that we had kind of connected. but this is kind of the first time that someone could measure in Excel bread. Since I would like to give you really short from every part a little bit, so I would like to summarize this therapeutic drug monitoring with this paper, which we published, I think, 2 years ago in Lancet, Infectious Diseases. So the idea is normally antibiotics are given by the bloodway, so which we called upstream and site of infection. So usually, of course, we would like to apply of infection on the tissue. For example, if you have a lung infection, we would like to target the lung, but we gave the antibiotics via upstream on the bloodway. So our idea was, would it make sense to measure the downstream, the fluid which is deriving from the infected area, site of infection? For example, in this case, breath. For example, if it's a skin infection, then is it sweat? So could we really measure in sweat, for example, the concentration? We have the blood concentration, and we could see the flux, how much is really going into the infected area at the end. So that's kind of, actually it was a hypothetical paper where we would like to combine our work with the wearables so that you could have either the blood, or you could use maybe ESF as a blood counterpart, as a upstream, and your wearable approach like breath or skin sweat measurement as a downstream. So what we do with this are polymer-based sensors, and maybe some of you heard about me because of that, so is the CRISPR-powered diagnostic. Now we are switching from antibiotics to nucleic acid, and with that we started in 2017. We were interested to measure microRNAs, which are actually could give us ideas about different diseases only if we measure many microRNAs, which we call the family of microRNAs, and see whether they change upscaled or downscaled. So that was our starting point. We had some different works, and at the end we came up with the CRISPR that it might have helped us to measure nucleic acids. So before I start with our approach, I would like to give you a short overview. So we have this CRISPR-based diagnostics. You can use two different modes. Either you can use a deactivated Cas enzyme, which can just recognize. For example, you can look for mutations in the whole genome, and if they are existent, the whole genome binds, for example, on a graphene field effect transistor and cause a charge change, because whole genome, of course, has a very much charges on surface, and so you could measure different genetic mutations without any target amplification. This is done by my colleague Kiana Aran in the UCSD in San Diego, and called as a CRISPR chip. So that's the first thing. However, if you are targeting shorter microRNAs, which are really 20 bases, so you don't have this effect in this case. So binding only would not help you. So in this case, you can go and use whole capacity of the CRISPR-powered diagnostic. In this case, if you forget the beginning amplification, if the target is present, the Cas enzyme gets activated, and for example, we have a fluorescent marker with a quencher. Once it's cleaved, so that you get a signal, and you could measure the target's concentration depending on the signal that you are getting at the end. So that was these two methods, and of course we choose also from our approach the cleavage-based approach. Maybe one side note: over the years there was a boom in the CRISPR-powered diagnostics, because as I said, I'm an editor in biosensors bioelectronics, and I don't know how much paper I have to handle because of this, because many people were interested. And this is not only because the discoverers got Nobel Prize and it was just hype, but because it's very easy to use and it is very versatile. So you have different enzymes which you can use, and you can target single-stranded or double-stranded DNA, you can target RNAs, and these are not only from actually coming from bacteria, but also Jennifer Doudna's group show that you can even get an Cas system from a bacteriophage, which is actually a virus. So this is, for example, this one also can target DNAs. You can see it's a little bit smaller. However, this one is not used for the diagnostic purposes yet. So maybe if you are interested, that might be a good starting point. What we did is actually electrochemical detection. So just to show you how simple is it, so you have your Cas enzyme, you have your CRISPR RNA, which you need to program your Cas enzyme for a specific target, and this is the blue part. I will show you a lot of more things on this slide. Forget everything. If you would like to target a new analyte, the only thing that you will change is this blue part. Everything else stays the same. We use also a reporter with a 6-farm and biotin, and then we labeled it for electrochemical detection. As I said, it's simple. You mix it in an epi, wait a specific time at a specific temperature, which depends on your Cas enzyme. And if you have no target, for example, microRNA in your sample is not getting activated, so you can build a complete assay, which generates you hydrogen peroxide, which you can oxidize and get a high signal. What happens if we have the target? So the CRISPR-Cas gets activated. I always saying a Pac-Man going through the epi and eat all the, cleave every reporter RNA. In this site, of course, the biotinylated site still can bind to surface, but signal is just decreasing. So we have actually a competitive-like a behavior, but I have to say it's not a binding-based assay, so it's not directly a competitive assay, but if you have a no target, you have a high signal. If you have a high target, you have a lower signal. And which we did it, the first work in 2019, so that was really the first electrochemical CRISPR-powered approach. And at that time, we had 4 hours for the whole process and got a detection limit of picomoles. So if you were interested in copies, so we are talking about million copies per microliter. So, of course, then the pandemic hit, and we started to work on COVID as well. And after 3 years, we will be able to publish our results. In this work, we also combined the antibiotic to the nucleic acid work. Actually, our approach was to show, look how versatile our system is, and we can process different assays on the same platform. So you can measure 6 channels at the same time. We use also some NFC potential state just to show how to increase the portability. And both assays, as you might also remember now, ended up with the glucose oxidase converting glucose into hydrogen peroxide. In all channels, we have the detection sites after each immobilization area, which means there is no chance of a crosstalk because we have just a run of the fluid and do the detection. And the idea was, once you get tested positive, so the antibiotherapy will start, and we keep the concentration above a minimum limit to secure a super or co-bacterial infection in this case. So what actually done in this work much more beyond the COVID was, actually we could improve our limit of detection from picomolar in a femtomolar range, let's say 1,000 times, by cutting the time from 4 hours to 30 minutes. How we did this? So we changed the ortholog, Cas ortholog, which we are using, to another Cas ortholog, which known to be more catalytically active and likes to cut uracils. So we did actually change also our reporter molecule to just 20 uracils and could go from a picomolar detection limit into a femtomolar detection limit. We tested this with E gene and the P gene of the SARS-CoV-2, achieving in both femtomolar limit of detection, as I said, in 30 minutes. The group of Jennifer Doudna also shows, since we have actually a virus and a very long genome again, they said, okay, can we use 2 different genes in the same genome and increase for 1 target, so to say 2 targets by targeting them? We tested these as well, so with this you can even go with a superior limit of detection. However, we never tested this in real samples, so I would see this with a precaution. And at the end of the day, we are still interested to detect microRNAs, and these are shorter, so we cannot target 2 different microRNAs simultaneously. We did it patient test with 4 channels, so we had a control, a negative control with DRP or previous SARS-CoV-1, and 2 channels which for the signal, and we did 2 patient measurements which has a high viral load. So at the end of the day, we could show that the signal is dropping, however, not much that we are expecting, and that has of course a reason, because in a calibration curve, you have only the reporter molecule. However, in a real sample, you have not only the reporter, but also all RNAs that are present in the sample. So now we are working also with the microfluidics to activate with the sample the CRISPR and then bring the reporter to achieve the same sensitivity. Just to maybe summarize this part is, and also say, maybe with this femtomol, we are talking about 2,000 copies per microliter without any target amplification. So, and if you are interested in CRISPR diagnostics and start looking for a starting point, so what I could tell you is amplification-free is the right way, because if you would like to combine these isothermal amplifications, then I would rather go for the isothermal amplification, because you lose all the attractivity of the CRISPR as well. Just a little bit selectivity due to CRISPR, for me, does not justifies the use of CRISPR. So then the second point is multiplexing, where we show that you can combine with for example, with a drug, but you can also do it with antibodies and the nucleic acid. And what you could maybe also do using CRISPR reaction as a signal amplification, for example, an aptamer, which recognizes a protein and either starts or stops triggering and CRISPR reaction. So that would be a case. And of course, the other thing would be the wearables. So you could implement this CRISPR-based diagnostic systems into the wearable devices. There is up to date only one from MIT, from Jim Collins' group, with the face mask for detecting COVID-19, and a Chinese group has a microneedle patch, but I don't have that much information about this work. So and there are lots of different wearable systems, so it might be also interesting for you to start. So now I would like to lose some word about our crazy project, which is called Lab on a Bird. So we started with the ornithologists, so and our goal was measuring hormones, and the idea was what happens if a bird flies through an ocean, how they feel. So that was the starting point, and it was a very small bird at the end, as you can see in the image. So the reality always differs from the proposal. So our idea was to detect testosterone on a freely moving bird. And as I said, we ended up with a chicken. We used this jacket, which you can buy on Amazon. So this whole system was implemented in this device. So the idea was to put a line onto the wing and get every 2 hours 10 microliter blood, mix it with and do a basic competitive assay. So it's there on that side. There's nothing crazy. The system looked like, so we put the electronics inside. So this is the needle, actually, and we use some peristaltic pumps, bidirectional, so that we can control, actually, which also can work with air. So we can would like to clean the lines and also take the thing. What we did on the sensor side is actually we keep the electrochemical cell and divide it from the microfluidic area. So here we made a single line where we put the assay components and do the assay measurement. And this is, after each measurement, it's just turned and done. So if you remember old photograph machines, so it works in a similar principle with a similar line, so that which is just turning for each measurement, and then you can do the measurement in this case. So you can see here, of course, this should be done under operation, so the animal gets some anestheticum, and then you can see after the operation. So one of the things, of course, which you need to test is the tubing problematic. So is the chickens are starting to pick each other's tubing, And of course, is it eating? So it's in animal behavior. So if it eats, it feels well and safe. So that was the idea that actually is, of course, it's running something like this. But anyway, so it's doing well. So and after the measurement, so we took also the system away. This project is actually currently a little bit stopped because of my move to the Munich. And I have to say, everything works. So we can draw every 2 hours blood from the animal. The line is not, it's a whole blood, so it's not stopping. The problem is, of course, hormones is, we are talking around picogram per milliliter, and they are measured, extracted. So, and we do not convince the bird to fly with 1,000 RPM around itself to extract it. And also cannot use solvent, so that's maybe we will go for another target than a hormone which secures the measurement. But at the end, so you really could connect the system with a Bluetooth so that it sends the data and you can run the animal into the laboratory. So that was an invasive variable. So let's look at the second approach and non-invasive variable. So this is started when I was at Imperial College London. I learned the paper-based technology there, and I decided to make it in Freiburg in a way so that we say, okay, it's electrochemically clean. We did it, and at the end of the day, we had this sensor on our hands, and then we had to ask ourselves the question, okay, we can measure hydrogen peroxide, but what is about the application? So we were not convinced about the application. So, and there we had the idea, okay, paper itself is a hygroscopic, so it's never dry. So it gets the humidity of the air. So the question was, if I put salt, because for electrochemistry you need salt, on a paper, let them dry, and then exhale to it, and exhale enough. Can we do an electrochemical measurement on a paper without wetting it completely? So, and the answer was yes. So, and this opens up, it's completely a bad engineering example. Usually you need to plan it from other side around, but actually we came up to the idea at the end. So it's a sampling instantly and also measurement method. So you measure, the aerosols are catched in the paper, and you do the measurement. Of course, you could also make weight for the measurement and then let them accumulate and then do the measurement. That's also possible. But if you would like the real time, that also works. The good thing is, paper itself, it's a substrate for the electrodes. You can even put enzymes or biomolecules on it, so immobilize on it, so it's a matrix for them. And as I said, it's a solid electrolyte. So you put salt, you exhale, and you have created your, for the electrochemical detection. And the good thing is, it's a non-invasive sampling method. So you need to exhale. So there is no way around. And in the first work, we showed that we could measure hydrogen peroxide in a simulated breeding. And of course, one thing maybe which I need to highlight here, breeding is a very complex. So it's, I mean, to measuring in a closed environment while you are breeding means actually you have mechanical changes, you have temperature changes. So you exhale with 100% humidity with 35 degrees, inhale the room humidity plus the room temperature depending on the thing. So it's quite changing. So the idea was we need to differentiate. So we have two electrodes, one is sensitive to the analyte, the other one is not, and we subtract the signals so that to be able to eliminate these effects. For the hydrogen peroxide, it worked, but it was not that sensitive, so we choose another step. Okay, we said, let's go for glucose because it's much more concentrated. We can measure from exhaled breath. And here you can see also how is it built. So you can print wax, you can screen print it, silver lines, and then put your carbon and mediated carbon electrodes. And in this case, even both electrodes are symmetrical. And on one side you put the enzyme, on the other side you don't put any enzyme. So it's just subtracting it. This is something which we are actually constantly and now in Munich working a lot. But I would like to show a previous result, so we tested this also on a master thesis actually my student tested that, and what she did actually 2 days she came with fasting, and in the second day she eats some sugar in between, and she did some exhaling, and in the second day she eats something which you can see here with the black, so the sugar load. What we see here, but for the first time, actually, it's a critic. So we see that our signals are not stable. So Prussian blue electrodes with exhaling is not stable. So currently we are working on improving the material. However, we can say that we see a difference. At least we can detect some glucose residues. We are far away from saying that we could quantitatively measuring it, but that's first step. We could see on the same person in 2 days with sugar load a difference in exhaling. And this is, of course, something which we need to further work on, and that's we are doing. So this image is coming from 2020, 2020. Once Nature Biotechnology asked me, because our CRISPR work on COVID, and then I told them we are also working mask-based sensors, and this is actually a working prototype from that part. So with the sensor integrated mask at that time with a wire, of course, which is made in 1 day. What you can do with such a sensors, you can do metabolites by adding enzymes. So you can measure glucose, lactate from exhaled breath. What you could do also is pathogen detection. So you could really collect on the paper. Maybe you can use some subsample extraction, and then do the detection. And everything should be in a smartphone readout. Of course, that was before we don't know a lot about the system. Now we are working in a newer version, which we called our breath sensor, which should be done in an FFP3 mask, and where we would like to install the sensor into the cap, and with the electronics, so you have the electronics always in the mask, so you have the sensor in different masks, and you just need to change the electronics from mask to mask with yourself so that you could measure. And we are, actually this is the software that we are planning right now. So now with moving to Zoom, I have a little bit more resources to realize that. So this would be the first work. So we would go for non-invasive glucose measurement, non-invasive lactate measurement. For glucose, we would like to go more in the work with the clinicians. For the lactate, we would like to work with sports students, so that, because on that side, it might be interesting to improve their sports exercises by measuring their lactate values. So in general, of course, to be another luck that we had in this idea, once we did the first mask sensor, we said, okay, but who is gonna wear a mask? And that was before the pandemic. So, and we started to think, okay, can we do, for example, some under nose patches, in nose patches, because we have really good sweat sensors. So we just need to reverse engineer them, catch the exhaled breath, and then do the measurements into these patches. Or we had some crazy ideas as well. Could we do some intrahea patches, which is self-powered by using metabolites, or which actually I like, but it was already patented, unfortunately. Someone wrote word sensors into a vaping device patent. So what you could do, for example, with the vaping device is what I like. You can really inhale a drug, for example, for a lung disease. And let's say after an hour, you can exhale and use the sensor capabilities that you build in. And then for the next hour, for the next drug administration, if the concentrations are not fitting, so it can also be a closed loop system. So that might be possible. And as I said, since it's quite patented, so we put all the ideas into our paper, which we published 2 years ago, in this how we would do it for getting rid of, for example, saliva, etc. So with the effects of saliva. In the some last minutes, I would like to talk about our latest approach, which we called OptoAssays. So this is a dynamic assay, which you can control by changing the wavelength. So the idea is you can use phytochrom B and its interacting factor. And if you have 660 nanometer wavelength, then they are together, and 740, they are separated. So they can bind and release. And this is a reversible binding. So we are not talking a single binding. So what you can do, our idea was, let's do a wash and pump free assay. So we started with a rectangle, but at the end it gets round, it's a circle. So we have a sender where we have the competitors, and in the receiver we have this detection spot, which will be measured. And so, I need to do it in this way. So just to show you how it works, so we made it for a HISTAC, just a demo assay. So we had an anti-His antibody in the receiver, and then we have GFP with HISTAC. And so, of course, it's 660. Once we change the wavelength and add the target, for example, another protein with an HISTAC, then they start to compete. And the idea was, of course, to change the wavelength so that the free GFPs with HISTAC, again, binding back. Of course, we are aware of diffusion, so that's for sure, but we would like to see if it's theoretically work. And just to give you an idea how it looks like in reality, so we have, as I said, circles, and in the middle, so it's 660, nothing should be released. Here it is released, and then we have analyzed so that the signals are changing, so high in concentrated probes. And then we, of course, we need a black box for this, where we control via smartphone and via Arduino, and the different red and far red lights for controlling the protein. We did, with this system, we were capable of doing a calibration curve, which was with the rebinding, receiving in a limit of detection around 8 picogram per milliliter. However, as you can see, we have still high standard deviation, so this is something which we are now focusing to improve these actually reliability of the system, as well as going for real targets, like something that we would like to really measure. Just to show you a short example. So these systems are based on nitrocellulose, which we just cutting with a CO2 laser in our system, and everything will be then put in such a holder where we immobilize these single nitrocellulose papers accordingly, either sender or receiver. And then, of course, the sample is introduced. So we are actually usually it needs to be done into this black box, and then you can use normal ambient light. Then we have the system so that we send this for 740 to release. And you can see the samples here, then 660 for rebinding, and at the end for the GFP, we have the elimination. Yeah, and then actually the picture is taken, and then the analysis is done, so accordingly. So this is actually, we are here, could show the proof of principle, so that such a new type of system could work. But actually now we are working for a project to, looking for a project to bring this forward to enable. And of course, these optogenetic switches allows us also a lot more. So you could really bring something from A to B and bind it from another place. You can bring it back. So you can really play with this. So, so we are looking for different approaches as well. To look for that. With this, I'm actually end of my talk. I'm always the front person. Of course, there are lots of people behind this work and also many collaborations that we had over the years. I finish my talks always in a similar way. So science is about sharing information, and therefore you can find me or my group in different social media channels. And if you ask yourself what social media got to do with science, I have a talk in YouTube. Maybe I can convince you to use it. With this, I'm end of my talk. Thank you very much.
Thank you. Thank you so much, Jan, for this visionary and inspiring talk. Thank you so much. So the talk is open for discussion.
Thank you, Jan, for your very illustrative talk about your research in general. So I have a question about the treatment of the samples when you collect them in your devices. So many of them rely on biochemical interactions that can be, for instance, be changing during the transportation to the final measurement, either like, for instance, impedance spectroscopy or some other more sophisticated. So how is the final reading of the analyte is affected by this transportation of the device to the final measurement device? I mean, the other one.
Actually, it's a huge topic. Definitely sample preparation, and this we saw also in many projects. It's different. So for the polymer system, which we use actually for CRISPR and antibiotic detection, we have usually either a plasma extraction before in like a laboratory setting, or even actually in lysis and sample extraction. But for the bread one, it's a little bit different. In this case, what we observed is, for example, for hydrogen peroxide, if you cool it, the exhaled bread, and let it, for example, because you need to also wait to collect enough sample. So in this case, what we saw in hydrogen peroxide, that this is really actually kind of disturbing and going away over the time, decomposes, in water. So you definitely need, if you would like to collect it, so I would do, for example, either dilute it in 10 times PBS, so that it just gets into the right conditions. That we do also, by the way, by the animal tests. So we directly dissolve them into 1 to 10 PBS. And in our case, we have already sold on paper for the mask. So there, I think you don't need any sample preparation unless you would like to measure, of course, bacteria or virus in a genomic way. So you would like to really lysis, etc., etc. So this would be something else. But actually, on the example for what I told for the CRISPR by Jim Collins' group, they also made, for example, an origami-based device, which had some sugar berries in between. So they had really step-by-step these lysis step, and at the end, so going to their sensing system. So that's, but regarding sensors, it's, I think, one of the most important topics, how you sample and how standardized is it. So because we collaborated in case of an Alzheimer disease for microRNAs also with the hospital, and it was, the situation was really interesting because, for example, they said even the needle size that you are getting the sample plays a role for their studies for protein measurement. And we made with them, we had another problem, for example, in the microRNA. So they searched the literature, find 250 works and reviews, and said, these are the top microRNAs for the Alzheimer. Then they said, okay, that sounds a little bit strange. And they did their nanostring measurements, and they came up with 10 different microRNAs, which were even in this list at all. So this is, I think, something not only related with maybe false research, but also with how you are sampling, what kind of other types of diseases are existent on the patient. So maybe these Alzheimer patients has other diseases which you need to consider. which affects the microRNA. So it's I think in the sample preparation, it's very important.
Yeah, thank you, Jan.
You're welcome.
Thank you very much for the presentation. Continuing with the question, the sensitive detection of molecules or biomolecules, proteins, needs a target antibody, any kind of a market. It means that it depends on the environmental conditions, the temperature, humidity, also the signal to noise, respect to the detection. But there is a market sensitive. For example, in this case, you just say label-free detection by impedance spectroscopy. There are limitations, but
that's just a suggestion, by the way.
yeah, there is a sensor that is with the possibility of market sensitive. That means that you can detect salts, electrolytes, changes of the index of fraction. For example, in the case of OptoAssays, you need also any kinds of enzyme or let's say some fluorescent molecules to be detected. The question again is there any on-market sensor, for example, or label-free sensor that can use in order to avoid all these environmental things?
Okay, if I got the question correctly, so regarding label-free, it's just a suggestion here. I think in general, label-free methodologies are really problematic, especially if you go like such a case, like breeding, because this is constantly changing environment, so this is kind of almost impossible maybe. But regarding the laboratory, more controlled environment, I believe that might work. However, if you leave laboratories, it's always the question, is the environmental conditions, how much they affect. So in that case, most probably labeling or like fluorescence or electrochemistry, kind of redox labeling would help. And regarding the market, so I think with this we are, I would say, far away from market. So because bread is different. So because, for example, in the first study where we measured antibiotics from exhaled bread, we would like to be sure, and we had also some animals which had some lung injuries, because the question is, okay, you would like to do bread diagnostic, but what about your patient? Does the lung blood barrier works correctly? So you need to be sure to be saying, okay, I would like to use it for diagnostics. So there are lots of parameters. And for example, we tested also on the animals with lung injuries and see that these nice correlation disappears or changes according to either the blood, blood carriers are growing or coming narrowing, so that has an effect on how this transfer works. So there are lots of issues and we need to be understanding. So what we aiming here is to improve the sensitivity of our sensors, and I think glucose or lactate is a good example because they are in a high limit, because in exhaled bread, so in blood you have, for example, in milligram. And if you look at the exhaled bread, so it's a lung fluid is 1 to 10, but then is this air plus aerosols is 1 to 1,000 or 1 to 10,000 dilutions. So we need to work more in this case. So therefore we just said, okay, we would like to finalize a stable system with electronics, and then we go with the sensors and also sensing materials, how we improve the sensitivity of our systems. I hope that kind of answers your question.
Thank you very much for your answer.
Welcome.
Any questions from the students? Please don't be shy. Yeah.
Thank you for your talk. I think some of the most interesting options for diagnostics is microRNA, but microRNAs are usually found in extracellular vesicles, and their concentration is very high cost. Are there any options, new alternatives to avoid this long time and cost for obtaining extracellular vesicles?
I think there you need to only work on the extraction, I guess. How do you get the microRNAs out of them? If I'm correct regarding the detection time, it would be 30 minutes. So it's really, the cleavage takes now 5 minutes, and the sample to result time is, in these sensors, we are in 30 minutes. So excluding the sample preparation.
Because in extracellular vesicles, I've seen they concentrate through ultracentrifugation or like that.
So
ultracentrifugation they use.
So we need this. So, but actually, usually what we use is 100 microliters columns, and you get the end, you get also 10. So you have already also some kind of concentration, concentrating it them as well. So we need to check. So this is actually something which we don't go this direction. So we were mainly interested in free microRNAs, but this is definitely also a thing which might be, most probably it will be conserved much better.
Thank you.
You're welcome.
Thank you for your presentation. My question is, how do you select the molecule to be detected? I mean, based on what? Because you have some microelectronics there, so of course there is a limitation in the electronics. But which is your the properties or any characteristics of the molecules you detect? How do you select the molecule to be detected?
Actually, we don't. So we ask experts. So this is actually, usually we are going to the doctors or clinicians or sports people, and then ask them what you would like to detect in case of like a mask. So for example, with the lactate and this exercise control came from their side, so that will be interested. So with the glucose, we have also nice ideas, because usually you can really do a calibration on a human, so you could really adapt the glucose concentration, measure it on that side, and validate with a mask would be a nice option. So there are mainly actually, we have of course some targets like microRNAs that we were from the beginning on, they were fitting also to our system. You cannot detect one microRNA and say you have this disease. You need to detect at least a family, which has around 6 to 8 targets. And then you need to, they were really appealing for our design system to say we have 8 analytes which can detect simultaneously from the same sample, and we could kind of get a pattern and maybe say about something about a disease. So this is kind of how it works. And usually what we learned over the years is really don't believe the literature. And if you are working on microRNAs, your clinical partner needs to test on real samples which microRNAs they really could detect. And we choose the top of them, the high concentrated ones, of course.
Okay. And also, any idea about the price for the electronics you normally use for detection? A range of price?
I think on the mask, I could say now, so it will be most probably 5 to 10 euros. Which once you have it in a big style, but for the other ones we still did not decide on a system. So because actually all electrochemical life has changed. Some couple of years ago, once analog devices decided to make a potentiostate as a circuit. And they now have, let's say, 4 channels, which are very small and very powerful. So now we are also implementing these systems in our devices. And so its price is really defined. But anyway, so this is actually the non-disposable part of the system. At the end, I think which will be counting is the disposable part. So this should be low cost.
Okay, and what about the disposal of all of this? Is it toxic for the environment? Is it easy for disposal? What can you tell about the contamination? Because you have obviously human substances there.
Yeah, so you need to recycle. I think, well, that's a good question. Once with human samples, I need to check if you could really recycle them. But for the paper-based ones, you can throw them and then so they will dissolve. Maybe you need to be careful because of the silver content if you do it too much. So there are also some ideas. So we are trying to also improve these things, but that would be definitely still a problem.
Okay.
For sure.
Thank you.
You're welcome.
Any other question from students?
I think there was a question on top. Yeah.
Yeah. No?
Okay.
Okay, so thank you. Thank you so much, Jan.
Thank you very much. Thanks for coming. Thank you. Thanks for joining.